Hello, everyone. We will be discussing cardiovirus, a group 4 virus. Our team consists of Alondra Lopez Brajas
and Kiana Tom.
This slide gives a quick breakdown of how we will be discussing the material in this video.
So about the virus, cardiovirus is in group 4, which includes positive single-stranded RNA viruses. When we previously studied picornaviruses, we studied poliovirus as the prototype. The virus we are presenting, cardiovirus, is also within the Picornaviridae family. Cardiovirus is the genus. It used to be divided into 2 species, but it was reclassified into 6, which includes cardiovirus A through F. This table shows the species, virus names, and the hosts. There are over 30 species of hosts, including humans, mice, and other mammals, birds, and invertebrates. Cardiovirus is non-enveloped, icosahedral, and approximately 30 nanometers in diameter. The genome is linear and has a single open reading frame that encodes a polyprotein. This slide shows the structure of the capsid and the genome with one open reading frame. Here is a close-up of the genome, which encodes the capsid proteins and non-structural proteins.
As mentioned previously, cardiovirus is a part of the Picornaviridae family, so we will see that its replication cycle is very similar to other Picornaviridae viruses, such as polio. Here is a slide that we are familiar with. It depicts the replication cycle for most cells. We will begin by discussing the early stage. Different species of cardiovirus have different receptors, and these receptors vary by host. For example, the receptor of EMCV in mice is VCAM1. Upon detecting a specific receptor, cardiovirus will bind. Binding will trigger a change in configuration and inject the genome into the cell. Next, we'll move on to viral replication. As mentioned previously, the genome is mRNA, thus we will use RNA-dependent RNA polymerase. Cardiovirus does not contain a cap. Instead, it has an iris. The iris will recruit the ribosome for translation initiation. There is only one open reading frame, so there will be one polyprotein that is produced. That polyprotein needs to be cleaved into smaller proteins. 2A, a protein within the genome, will initiate the cleavage. We have seen in other Picornaviridae viruses, such as polio, that 2A also cleaves EIF4G, which results in only viral mRNA being translated. When this occurs, the host mRNA is no longer translated. However, 2A does not do this in cardiovirus translation. More cleavage occurs, ultimately resulting in RNA-dependent RNA polymerase. As the Picornaviridae virus, cardiovirus has to bring its own polymerase. The RNA-dependent RNA polymerase will begin transcription. First, it will create an antigenome, which is useless to the virus. It will take this antigenome and transcribe an actual genome that can then go on to be translated. Once translated, the genome can move on to the late steps of the viral cell cycle. This slide shows the genes that make up the capsid proteins. These structural proteins will be cleaved and then assembled into a procapsid. The genome will be inserted into the procapsid, creating a provirion. A final cleavage of VP0 will transform the provirion into the virion. This virion will replicate until lysis occurs, releasing the virions from the cell to go infect more cells.
So now we will cover some of the diseases caused by cardiovirus and the available treatment. Some of the key diseases include myocarditis, encephalomyelitis, multiple sclerosis type symptoms in mice, reproduction disorders in pigs, and type 1 diabetes in other animals. We will cover the first 3. Myocarditis is inflammation of the heart muscle, which can cause rapid or abnormal heart rhythm. Symptoms are listed on the slide, and the treatment depends on severity. I wanted to mention how they found one of the cardioviruses, EMCV. There was an adult gibbon who had died of pulmonary edema and myocarditis. From that host, the researchers were able to isolate EMCV.
Cardioviruses can also cause encephalomyelitis, which is inflammation of the brain and spinal cord. The virus causes demyelination in those areas. The symptoms are listed here, and the treatment includes antiviral medication. Cardiovirus has been known to cause MS-type symptoms in mice. In MS, the immune system attacks the myelin of nerve fibers. The symptoms vary depending on the severity of the nerve damage, but can impact movement. Numbness, weakness, and tremors may also be present. There is no cure, but it is possible to slow the progression and manage symptoms.
So now about the vaccine. There is a vaccine used on pigs and zoo animals to prevent disease. It contains an inactivated form of the virus. This is analogous to the Salk vaccine, which was used against polio. And finally, we'll end with epidemiology. Cardiovirus can be found worldwide, as we previously mentioned that there are over 30 host species. Significant research studies have been performed in Siberia, Peru, and Pakistan.
We have seen outbreaks specifically in swine, mice, and elephants. An example of zoonosis occurred in New South Wales, where an infestation of mice resulted in the transmission of the virus to pigs.
And so this slide summarizes what we talked about today. So thanks for listening to our presentation.